I'm Nicole Theodosiou, and as chair of the Professional Development and Education Committee, it is always my biggest honor to introduce the winner of the Viktor Hamburger Prize at the annual meeting. The Viktor Hamburger Prize for Education recognizes individuals who have made outstanding contributions to education and developmental biology or related fields. And I'm thrilled to introduce this year's winner, Dr. Christopher Wright. Dr. Wright is a professor and director of the program in developmental biology at Vanderbilt University. Numerous former postdocs and colleagues spoke of his genuine caring for trainees, encouraging them to succeed to the best of their abilities, his cheerleader attitude, and his unflagging enthusiasm for science. He's quick to turn mistakes around with a, quote, "Well, at least we found that out early on," or, quote, "Don't worry, you'll never do that again." In his own lab, he has mentored over 14 graduate students, served on over 120 graduate student committees, demonstrating the respect that students have for his mentoring style. We are honoring Dr. Wright today for his crowning achievement in developmental biology education and training at Vanderbilt, the NIH-funded T32 training program that he designed and has directed for over 20 years. Since taking the lead role in 1999, it has successfully been renewed 5 times, highlighting his devotion to graduate education. The T32 has supported over 60 predoctoral and postdoctoral trainees, and importantly, he invites faculty and master's level students from nearby Meharry Medical College and Fisk University, historically Black institutions, to participate fully in all of the grant training activities. Many of these students have gone on to join the Vanderbilt PhD program, and it's because of Chris's mentoring and encouragement that gave them the key desire to be part of the program. So it is my honor to award Dr. Wright the Viktor Hamburger Outstanding Educator Prize.
I took a different tack on this talk, and I chose to sort of go at it from where I am inside my brain all the time, which is I am constantly excited about developmental biology. I just think it's an amazing thing. Every time I teach Xenopus embryology and we do an in vitro fertilization and I see thousands of eggs go to the 2-cell stage together, I can't barely control myself. So I think that there is an incredibly bright future for developmental biology, and we've gotten so far with various aspects of analysis, etc. But I do think it's time, and I hope you don't mind, but I'm going to sort of take a step back a little bit and try and propose some ideas that it's about time also to take a slight sort of kick in the pants ourselves and say, oh, we've gotten this far, surely there's some remaining challenges. So that's with this sort of spirit of me being super excited about where we can go. And hopefully before I die, we will have a blueprint for one organism and understand everything of how everything works. So sorry for being a little bit kind of humorous there. I'm really, really excited to be given this award in particular because, as I've shown you there, it's just following on from some people I regard as just amazing biologists, right? And so I felt sure they got this award wrong. But these are people, you can see them there, Sean Carroll, Lewis Walpert, Scott Gilbert himself, over there, our great Ida Chow that everyone knows and loves, and she just runs everything so perfectly. But so to see myself coming to this, I was really, really thankful. My first office, my enormous thanks to Dr. Maureen Gannon, who was, as you see, their next postdoctoral fellow with me, and she was the organizer of the nomination, and also thanks to the other letter writers for their outstanding support, and the selection committee members. I was extremely surprised when Olivier called me on the phone. Absolutely shocked, but wowed and grateful. I think this indicates that there is extramural recognition. Often one finds it's quite difficult inside your own place, especially with the higher-ups, to understand that they get it, that you're trying certainly scholarly pursuits, and to be recognized outside certainly made a difference where I am at, and I'm going to leverage the hell out of this. while our individual technical conceptual contributions will likely be submerged under a tsunami of data and discovery, and I would say that, interestingly, specific concepts, I've been in biology long enough, 4 decades or so, that they seem to undergo rediscovery every decade or so, with a new name or a new acronym to describe it sometimes. Take all this humorously, everybody who's inventing new seek names. I think that the training and mentoring impact at all levels will endure, and it's what I now focus most of my energy and excitement in. It's really going to young faculty, intermediate faculty. When I say young, I mean junior faculty. I'm sorry for saying young. And also trainees of all levels. I think that there are ways to prevent stagnation and to maintain at the same time that discovery science, especially developmental biology, is completely fascinating. I think we have to do a better job of cross-pollinating between the clinical and basic science bases and with other fields such as physics and chemistry and computing, and I'll have a little bit more to say about that later on. I've got my acknowledgements here. I thought it was okay to do this. I feel really uncomfortable doing it too much, but people I've trained with have just really set me up in a very privileged position. I came from a very blue-collar family. I was the first in large family to go to college, and I was very fortunate to go to the University of Warwick in the UK. And Hugh Woodland and Alan Coleman, Liz Jones and Bob Auld are the people that taught me during undergraduate, quote, biochemistry, unquote, but we got to see tadpoles and saw plasmids that had been injected into early embryos and that could produce casein inside of tadpoles. So that's very forward-looking to go to a university in those years. And then John Nolan, my DPhil supervisor, don't worry that the DPhil has the same quality as a PhD. I do have a PhD equivalent. This really taught me self-sufficiency. There's a lot of reading between the lines there. And you can also see very notably that there is a Gurdon connection, and I feel really, really privileged again to have come up through this. Eddie de Robertis was a postdoc with John Gurdon, and he was a terrifically supportive postdoctoral mentor. It began a 5-year postdoc with him. My interview with him was in the King's Arms in Oxford, in the center of the city, which was a great interview, I tell you. And now I've also put something in here about the people that have helped me a lot over the years. Bridget Hogan recruited me to Vanderbilt 33 years ago. It's really fundamental to find strong advocates and candid advisors and people that you can believe in 100%. And her role in my career has been 100% how I got here. And there are other people that I've listed here. I won't go through this list. These are partial listings, as you can see. And the bottom one there, I'm really sorry, because there are people that I want to mention, like, I'm going to say LSK, because I know she hates that. Lila Sonichikaso, so she hates being called LSK, right? Okay, and then Bruce Apple and Billy Swallows here, and lots of people that we've really had great time with. Okay, so here. I hope many of you have read this from Claudio. How many people have read this? Some people have read this, but not everybody. Everybody should go read this. Even if you don't agree with it, it makes you think. So he has got some ideas in here, and he comes up with Claudio's position. You could imagine dour Claudio. The picture is of dark clouds on the horizon, and it's a call to arms to try to regain what the field has been losing. He has this kind of abstract, which is developmental biology is in crisis. You can read the rest of it for yourself. But he says that some new technologies encourage big data generation at the expense of uncovering principles, and that most fundamental questions have not been answered at the end there. I've added my post-it note in there, or even posed correctly. One of my goals in education is to get people to ask the right question in the right way, using the right words. And I really struggle when it's clear, as I've written there, that there is a difference between hypothesis and prediction that some people really don't understand. Now, I'm not saying that we are necessarily at the sorry state that is potentially indicated within Claudio's article, but I do want to say that I think RO oneness and other threats mean that we are sort of could be, I agree with him in one way, could be peering over the precipice. And my personal view is that the R01 is stifling inventiveness as compared to, let's say, the Mira R35 in the US system. I've been chair of both of those types of panels, and I think the inventiveness inside the R35 panel is really quite stellar. I would just use this moment on the soapbox to say that the $250K modular budget is plainly ridiculous, and we need to get that out of the way. Okay, so a smidgen of science. That was enough pontificating for a while. Smidgen of science. We've heard several times, nothing supplants watching cells behave in real tissue in real time, alongside what I would call deep dive molecular analyses with precise correlation. So spatial transcriptomics and cross-referencing between single cell analysis, and then using some marker genes to using the spatial transcriptomics to put the cells in place, and now maybe moving forward and doing beautiful techniques like watching things in real time. I think this is the way forward. I think that new questions and new cell biology often emerge. I'm going to show you just one example from our own work here. This is a pancreatic explant taken from the mouse, about mid-gestation, and it shows you that active epithelial aggression is being held back by apical lumen tethering. So we heard early on in the conference about how cells break through a basal lamina and how that is a control process. This is a cell that will not break through a basal lamina, and it's important that it doesn't break away and disappear. So it's a really important process because the cells have to stay together near each other to cluster into the islets of Langerhans, which have purely essential endocrine function in maintaining glucose homeostasis. So if I play this movie for you, and you've got the details of what's going on down below, what you'll notice is that the yellow arrowhead is pointing out the apical tether, which stops this cell disappearing through the basal lamina, I believe. And so we did this, I should just back off a little bit. We sat back about 6 or 7 years ago. When I say we, it was me sitting in my office and looking at papers and saying, there's something missing. We don't understand organogenesis. It's all a little bit kind of frou-frou. And so let's go back in and do some really good descriptive work. We made some fantastic tracers, and we were able to watch the cells. What you're seeing here is the nuclei of endocrine-committed cells that are egressing from the epithelium, and they are marked with red for the nucleus and then green on the membrane. And you should be able to watch now these cells, the apical tether that's pointed with the yellow arrow there. You can notice that there's a lot of filopodial protrusive activity, and cells that are younger are just starting to appear in this area now. Cells that are younger are coming up. All of those cells can be seen to have an apical tether. So it's really important, actually, that we can watch those sort of things, because we came up with immediately, and I'm just highlighting a couple questions that came up. First of all, the old model was that cells would break through after egressing the lamina, would break through and disappear in what I call the space shuttle launch mode, and disappear to a faraway islet. And this is completely untrue. They can't do that. What happens is the basal lamina becomes weakened and distorted. There's no breakage. They swell the basal lamina, and other cell types are coming in from the outside. So this is what creates the complex mixture of neurons, endocrine cells born from the epithelium, and endothelial cells that have to come in to make the capillary bed for which the blood and endocrine cells can exchange information. So we came up with immediately several sets of questions here, which we're still trying to answer. But that is new cell biology, and we took, by taking that step back and saying how true is the literature, we were able to sort of, I think, create a whole new set of engineering questions. So I'll go back again to some more thought processes here. I think research, discovery, and developmental biology is constantly amazing, beautiful, and satisfying. And these are the things I think about if I was a junior investigator today. And so we've started having some of these thought exercises. You can't run this as a course because you can't have an exam at the end because there's no truth, there's no answer to this. But the thought exercise is to have a free-flowing discussion which says, unlimited by technology. And you're not allowed to say, machines can't do that, or it's too expensive. But unlimited by technology, what's the singular most significant gap in your field right now? And what must you know or do to fill it? And then the idea is that you get people to start thinking bravely again, which is that technology might develop, and you might even develop that technology, or you might innovate a combination of techniques to allow you to answer or fill some of those questions. And I think it's really important to start thinking about lateral thinking and to be informed by being voracious about all areas of biology. I go to talks or I listen to the physics of microtubule assembly, the pure physics of microtubule assembly on glass slides, because it could be important to me as a developmental biologist. So I think another concept here that you've heard multiple times, but I'm just going to emphasize it because it's important. Pay attention and think deeply. There's nothing in the literature which is other than the best current model. I do have a view now that reviews are of limited use to me. I think the authors often seem scared to offer opinions rather than just sort of correlate and collate information. I think be as brave as possible. I'm talking to the trainees now, but don't jump forward just because your PI told you. Think about these things overnight or longer. Think long, hard, and continue talking it over as you start the experiments. I think in the field we need curated, integrated, and synthetic views about how developmental machines run and are influenced and how they become disturbed or break. And to trainees at this meeting, I want to reveal two almost complete career failures that I went through, because I think we need to tell people that imposter syndrome or an equivalent exists at all levels. It's part of being humble, I think. As a grad student, I could not get scattered plot binding assays to work to purify the steroid receptor I was trying to purify. And the F and B means there was much effing and blinding during those few months. I finally read a lot, and I got in with the tissue nuclei and proteins, and suddenly it started working, thank goodness, because as I told you, John Nolan, my PI, told me that if I didn't get the assay to work, I wouldn't be getting a DPhil. And as a postdoc, I couldn't get ECOR1, many of you don't know what this is, but I couldn't get ECOR1 sticky end plasmid construction to work. I couldn't affinity purify homeodomain protein antibodies. And the success of these last two things were actually what founded my independent career. I couldn't get it to work for months. But my goal then was to contact people doing similar stuff in Drosophila. And finally, yes, it worked. And I led to lots of publications, plus also, as I wrote there, a big bear hug from Eddie de Robertis, and Englishmen are still uncomfortable with hugging from their PIs. It's a little somewhat counterintuitive as I go into the next part of my talk, and somewhat backward from my privileged, successful developmental biology faculty position. And remember, some of us grew up as molecular developmental biology developed, and they were marinated. I was marinated in the progressive discovery from various models. And it's difficult, but not impossible for current trainees to go through this. I think you need to find people who can help you navigate. It's not just information overload. There's this way through it. But only now, it turns out, am I finding the time or the interest at some level to read back into the history of the Society for Developmental Biology and also DevBio in general. We know we've got some favorite modern texts on this topic. I bought every single one of Scott's developmental biology editions, and then when Michael Baresi and Scott carried it on, it's fantastic. Thank you guys. I also love the Principles of Development book by Lewis Walpert, and then also Cheryl Tickle. But I was gifted the Spemann and Waddington books out of the blue by Dr. Lauren Hoffman, who was a reproductive biologist and scholar at Vanderbilt. That is a 1938 edition of the Spemann book, and it's a very early edition of Waddington's. So it allowed me to start probing back into really some of the history of how developmental biology started, or at least the way, again, remember, I'm out of the school of Gurdon through multiple people, the way that things have developed. And preparing for this talk, I just went online and I searched for educational challenges in DevBio or challenges in DevBio. And I came up with this report. This is a letter from, I'm going to try and read here. It's Michael Locke here, who's the editor of this book. This letter is from, you can probably read it better than me. Can you read it? Okay, good. So in here, this is funny actually, because this is the issue. This letter is talking about a book that just got published, but I had to snag this out as well, because the developmental biologist, since he is in no danger of distracting the biochemist from the search for gene repressors, can with a clear conscience continue his harping on some of the complexities of the business. And I have friends of mine who think that I harp on, so I will move on. In this book, and I would tell you, get your PI to buy this book. It's available for about $50. This is the 25th Symposium report from the Society for Developmental Biology. It's fascinating. In here, it's a great introduction written by Jane Oppenheimer, who's unfortunately dead now, but she wrote about the first meeting of what used to be called the Society for the Study of Development and Growth, and that's the first annual meeting of that. Now, we're a bit bigger than that. That's about 30 people, maybe 40. So I understand some of the things I say might be difficult to implement, but I still think we've got to try and get to it. Okay, so one thing that she said that struck me was, and I've highlighted it here, how many selections of speakers made by the society have been other than relatively obvious? And then how many of the speakers would, like one person she drew out here, have been surprised to be invited? So I think we've got to try and think about, at Society for Developmental Biology annual meetings, inviting pure physicists, information transfer people about what are we doing with the slew of data which is coming up. So I think we need to interact with other people with distant fields in physics and chemistry, and I ran upstairs and I added this in there. Hernan Garcia, fantastic job today. That was an amazing talk. I think we need to interchange detailed information between studies on various organs. We need to find how to curate and synthesize overall knowledge. Dynamic details are important, and we have to be inventive. And I'm not being silly here. I think that the video game coders are really interesting. They know how to handle massive amounts of data and port it between different systems. And so I'm not suggesting a silly thing here, but it does require money and it does require people. I want to know, for example, considering gene regulatory networks, where the weak links exist and where weakness is introduced in human disease or developmental syndromes might be, and the push or pull points for productive, controllable resetting of the GRN. There are things that I've started reading about here. This is from the Allen Institute, a computational challenge to help developmental biology. I'm putting these in mostly for the trainees because I would recommend that you start reading in these areas. I think these are where the frontiers are. And then also deep learning for bioimage analysis in developmental biology. This is a nice review here. So couple of closing comments here is that if you look at cell states, then probabilistic distribution or cloud states of cells, this reminds me much of electrons in orbitals around atoms, which are stochastically arranged, in which it's hard to predict position and other qualities, and there are unallowed and allowed transitions. There are even things like quantum tunneling. And I think we have to start thinking about cell space as compared to chemical space. How is cell space explored? Can we controllably extend natural cell space? And are there quasi-cell states? And do or can cells behave similarly to electrons, is what I mean there. Are there ways to discuss such issues practically and proactively? I think that we should possibly, I'm just suggesting an idea, that plenary sessions at the SDB annual meeting could have everyone involved, or at least present, to identify new big challenges in developmental biology. One for me is carbohydrate modification of proteins, in the extracellular matrix especially. Really hard to think about how to get into that, but it's a big challenge. So I'll leave you with my final slide then, which is just thinking about knowledge as areas covered by lily pads. And I try to leap from one lily pad to the other as I navigate through my scientific career. But it's really much more informative, I think, to understand the inherent linkages underneath. So that's a top view of lilies, and then an underneath view of the connections which actually allow those to emerge. So I'm thinking that we can understand some of those conduits between these lily pads of knowledge, and I do see a very bright future for developmental biology. Thank you.